Hi there, I'm David Agard from UCSF and the Howard Hughes Medical Institute. And in this lecture, I want to talk about going beyond the diffraction limit using conventional microscopes. And it's a methodology known as both structured illumination microscopy as well as interference microscopies. And we'll be talking about both these in terms of what their potential is for biological imaging. So as you all know, that the fundamental limit in light microscopy is the wavelength of light in the numerical aperture of the objective lens. And what we see here is a cover slip, an objective lens, and this angle that defines the maximum ray that can go into the objective lens really defines not only the light gathering power of the lens, but what the resolution is. And in a previous lecture on deconvolution microscopy, we talked about the point spread function and the details and the mathematics of how resolution is defined and what the consequences of this limitation are. But now what we want to talk about is how do we go beyond what seem to be these inviolate physical principles of both the finite wavelength of light and the finite numerical aperture of the lens. And effectively, what there are is there's two methods where we can cheat physics, and that's what we'll be talking about in this lecture. And the first strategy is to collect more of the information. So why use just one objective lens? How about all this light down here, which is being lost? If we can capture that, we should be able to capture the information from that and effectively have a much higher NA coming from the lens. The second strategy that I'll focus on is using structured illumination to improve resolution. And that is, as you'll see, instead of illuminating the sample with a uniform field of light, we'll actually put pattern in that and look at what the consequences of that are and how we can then use a computer to extract the higher resolution information. Okay, so as we talked about before in terms of what the transfer function of a microscope is, I now want to talk about what happens when we have two objective lenses. And so as you imagine in this simple microscope, which now has the sample wedged in between two objective lenses, what we do is we illuminate with light that comes in through one objective lens, but the emitted light that actually contains all the sample information goes through both lenses, both that one and this one, and comes around this little prism and interfere with one another before being collected on the CCD. And that interference pattern will be a way of finding out much more high resolution information about precisely where the light came from. Because as you can imagine, only light that really has a balanced path between this path and that path ends up constructively interfering on the imaging detector. Okay? And so the way we can think about that is here's the light field that's coming out of the sample or going into the sample. What makes it through the objective lens is just this bit that will make it through angle alpha, as we see here, this wedge part. And then the total information that we can see is the convolution of this with itself, because this is the wave and what we measure as intensities, and that's the self-convolution. And that's this large region of support, like in the XY plane. Okay. But here, when we have this convolved with itself, we get this pattern, which is what we showed before in this donut-like toroidal structure in the deconvolution lecture, showing what the three-dimensional imaging profile is of a light microscope. But if we have two objective lenses, then we have this convolved with itself, which obviously contains way more information than this. And you can see that we come much better at approximating the full information field that we would have had if we had essentially an infinite numerical aperture for our objective lens. It's still limited by the wavelength, which defines this circle, but nonetheless, we get to collect essentially all this information, providing much higher resolution information. Okay? Now, this is what you get, it turns out, either whether you illuminate through both objective lenses or you observe through both objective lenses. But wait, there's more. We can do it. We can both illuminate through both objective lenses and observe through both objective lenses. So we create interference patterns for the light going in, and we create interference pattern for the light coming out. And that does even more for us, as we can see here. So here is that simple microscope where the light comes in. It goes through one objective lens. It also goes through the other objective lens. Similarly, the light coming out of this lens comes back up and then through this dichroic goes to the detector, and vice versa, and as well as through that one. Okay, so through both paths, and the result is not just this, which I showed you before, in terms of kind of coming up to the full circle of comparable resolution in the z direction to the xy direction, but you can now get double the resolution in the z direction than possible in the xy. And so you can get very, very high resolution information along the axial direction, which was normally something very badly behaved in any kind of conventional microscope. Moreover, you see that all the missing regions, both the missing cone right here, and then these missing regions if you just illuminate or excite through or look at the emission through both lenses, and you can see all that gets filled in very nicely. So there's a huge increase in lateral resolution, and now everything is observed so that we don't have to try to use very powerful methods that recreate the missing data. We can just deal with the fact that this intensity is lower than this intensity, et cetera. Okay? And the result is here. So this is, for example, a single fluorescent bead imaged in the xz direction. So this is the focal direction. You see the characteristic elongated pattern that you get for an as the out of focus light accumulates as you go up and down. Now, if with the two lenses using both the detection and illumination, what you see is you get this complex pattern, which is an interference pattern, but is basically shows very sharp detail, especially in the z direction. And then using either the Wiener filtering that I talked about in the deconvolution section, where you just divide by the intensity differences, or an even more powerful iterative deconvolution approach, you can see that you can get to very high resolution, where the possible resolutions now are like 70 nanometers in the z direction instead of something like 600 or 700, which would be for a conventional lens. And it's actually much better in the z direction than xy. Okay, and so this just gives an example of a cell, a HeLa cell, where the microtubules have been stained with fluorescent antibodies. And then what you can see is here would be if we took this image, a normal XY set of sections, and deconvolved it, we see we get rid of a lot of the blur, but nonetheless, that's a radical difference from what we get using this two-lens interference approach. And now you can see very fine detail throughout the entire depth of the sample. Okay, so this is a very powerful approach. The kind of drawback from it is that you actually need to have your sample wedged in between two objective lenses, and everything has to be perfectly matched, and the path lens have to be very tightly controlled. And so it's an experimental nightmare at times, but the results are really quite extraordinary. And it's much more sensitive because, again, you collect all the light from both sides, and you get to utilize that in making the image. There's a 4-pi, there's a confocal version of this known as 4-pi imaging, and there's a commercial instrument from Leica based on this technology that does this in a confocal degree. But that is actually the confocal, the commercial instruments are more like this, where you're only doing one of these illumination directions and not simultaneous illumination and emission through the two objective lenses. Just because that's much more demanding technically. Okay. The other strategy, which is a much more, I think, universal strategy, and although both of these lend themselves to live imaging, is what's known as structured illumination microscopy. And in this microscopy, instead of illuminating the sample with a uniform field, we illuminate with a pattern, like a grading pattern. And the concept of it and why it does some good is shown here in this simple slide. And this is, many of you, I'm sure, have seen a moire pattern. If you take two screens from like your screen door and you overlap them on one another, you'll see these interference patterns where they constructively and destructively interfere. And really what that is that the pattern of one screen is not precisely matching the pattern of the other. Why we care about this is the fact that if we imagine that the screen in the back is the object, and this one in the front is the illuminating light that we illuminate with in a grading pattern, what we see is that the fringe patterns that we observe are at much lower frequency. They're much coarser than the actual screen pattern itself. And this provides a way of getting high frequency information that would be beyond what the objective lens could see and putting it back into the objective lens and capturing that. Okay? And so experimentally what we do is we illuminate the sample with patterned light. Actually, we literally use a grating, and we observe these interference fringes between structure in the sample and the interference pattern and the grating that we're illuminating with. Now, when you look at the image, it looks like you've made a crummy image that has all kinds of patterning on it. And so, but there's information in there from beyond what would fit through the objective lens. And so this methodology requires computers to reconstruct the image and extract all the information that's present and unscramble it so that we can actually make sense of it. And so I want to briefly describe that in a little more detail. So if we imagine that, here's the observable region, essentially the limit of what light can pass through the finite aperture of the objective lens, but there's all this other space out here, information, that is unobserved from our sample. Okay? And what we're trying to do is to figure out a way of capturing all this information and understanding it, because that's the high resolution detail that's lost by both the finite objective aperture and wavelength of the lens. And so when we illuminate with a grating pattern, it turns out that's equivalent in this sort of diffraction space, Fourier space, of illuminating with information that has 3 spots, one here, and one here, and where the frequency of that pattern is determined by sort of the radius of this spot from the center of the image. And this, again, is a convolution, just like we talked about before, but now this is the convolution, the multiplication of the illumination with the sample in real space becomes a convolution in diffraction space. And what that has the benefit of doing is it makes 3 copies, in this case, one for each of these. So effectively what we're doing is we're taking this whole information regime and we're placing it at this center, and that center, so that they all overlap. Now, when they overlap like this, what we see is that we get information from down here, comes through in the guide that's shifted up and makes it into the observable region. Information that was up here in the guide this one that gets shifted down also maps into the observable region. And so we have all this extra information that makes it into the observable region. But it's all added together. It looks like a bit of a mess. But it turns out simply by recording 3 images where we shift the image a little bit, 120 degrees, 240 degrees, 360 degrees, we can sort out those 3 components precisely just by using some simple algebra. And so that allows us to figure out how to extract from all this combined information here the information that belongs up here and belongs down there. Okay, so again, here's the observed region. The spots indicate the frequency of the pattern that we're illuminating with. And as you can see, what we do in practice is we try to make absolutely the finest pattern that can just squeak through the objective lens. It's right here at the limit of what will get through the objective lens. Anything out here won't make it through. Anything in here we can see, but it's at lower resolution. And so by making the finest pattern, we can get the most information. And what we end up getting is from the grading in one position, we get all the information here and all the information here, and we get to map that into the objective lens. And then we can repeat this by rotating the grading into different directions, 2 directions or 3 directions, and we can then do a very good job of filling up the space so that we now get out to twice the resolution. Okay? And turns out that was just sort of what we do in the XY plane, but we can also create interference patterns in the Z direction as well. And that has the benefit of not just extending the kind of axial, the lateral resolution in the XY plane, but also takes care of this missing cone region. You see our little, in this structure we see our little toroidal donut, but it convolves that out, and so we get much higher resolution in the Z direction, as well as filling in all the missing regions. Okay? And this is done very simply. The microscope is really quite simple. We have light coming in through a fiber optic, gets focused, goes through a polarizer, focused on a grating. That grading gives 3 diffraction orders, and these then get placed where the limit guys are imaged right at the limit of the back focal plane plus the central term. They then form structure on the sample in X, Y, and Z. You can see here's Z interference terms and X, Y interference terms on the sample, and then we record those images. And what that allows us to do is to build up these big regions of Fourier space where only this little, this donut here would have been observable with a conventional microscope. Okay, so huge increase in resolution in X, Y, and Z over a conventional microscope. And so by way of example, here's a fluorescent actin stained HeLa cell. You can see all the stress fibers in the cell under normal illumination. Down here is where we illuminate with one of the structured illumination patterns. If you look very closely, you can see little stripy patterns along the sample, which is essentially an interference of the diffraction grading with the actin filament structure. If we take the Fourier transform of this, we see the structure of the actin bundles, but with the when we use the structured illumination, now we see new structure, this little spot here and these rays coming off like that, and similarly down here, that represent the new information that was displaced really from outside of this circle that gets mapped into the circle that we can observe. And so when we do this, for example, we have 7 different components. There's a central component like you would normally observe, and each of the displaced components, this is done with 3 different orientation angles where you can see new components that have been reconstructed that go to much higher resolution. And when we put all that together, we now see we have a Fourier transform that extends twice as far as what would have been observable in a conventional microscope. And when you look at the consequences for an image, the results are pretty stunning. And so you go from an image like this to an image like this. And so clearly much, much sharper detail. You can see actin filaments that have gotten extraordinarily close to one another. Here's just a blow up of that. And so where normally you would just see one big blur, you can see completely resolved separate actin filaments that are very close to one another. Similarly, in 3 dimensions, this is just with fluorescent spheres. You can see instead of having the normal, horrible, out-of-focus blur in the Z direction that you would normally have, then you see that tightens up quite dramatically without having to do computer processing in terms of deconvolution, but just because we now observe much more information in the Z direction, and we see superb XY spatial resolution. Now, it turns out this method can actually be combined with that interference methodology I talked about in the beginning and actually get 3-dimensional resolution all the way around that's uniform at very high resolution. But here's just a couple of examples where you can see quite dramatic differences. These are neurons, and these are synaptic boutons from Drosophila neurons, and you can see the difference between the before and after using structured illumination. And this methodology lends itself very nicely to live imaging as well, and it's only limited by how fast we can form those patterns. And recent work, both at Genelia Farms and here at UCSF, has used solid state devices to generate the patterns, essentially like in a video projector, instead of having to have a physical grading that you rotate and translate. And that allows us to do a complete 3-dimensional stack within a second on live samples. So it's quite exciting. Here's just a few pretty examples of showing multicolor imaging using structured illumination. So here's chromatin in a cell, nuclear envelope being stained, as well as microtubule cytoskeleton. And then you can zoom into the nucleus and look at deformations in the nuclear envelope around the chromatin. And you can, in fact, if you look very closely in this, you can see the nuclear pores, and you can see where the nuclear pores actually go in and push the DNA out of the way, all with light microscopic imaging. Okay. Now, what are the limits, right? I mean, it would seem from this that the best we can do is a factor of 2 in resolution. And so the reason is, here's the observed region, and then we create a pattern, and then that allows us to access to information out here. But what if we could somehow create a pattern that had frequencies that were much higher frequency than defined by the diffraction limit of the objective lens? If we could do that, then we could get information about arbitrary sample information at any resolution you wanted. The problem is how do we ever create a pattern that has higher frequency content and can fit through the objective lens? And linear theory says that this is impossible, that you're limited. The best you can do, which as I showed you before, is you put the pattern, illumination pattern, right at the limit of the numerical aperture of the objective lens. But it turns out that there are strategies where we can exploit nonlinear processes of the dyes themselves to get much higher resolution. And two approaches have been used. One is to actually use nonlinearity in the photo response of the light of the dye itself, or to use photo switchable dyes where we can turn them on and turn them off, and the turning on and off then is an inherently nonlinear process. And just by way of illustration, here is an example from trying to do this with just by cranking up the excitation light until now you get a nonlinear response. That is, here's the illumination, here's the emission from a fluorophore. This could be from any fluorophore, it doesn't matter. And that at low illumination levels, you get a very linear response. So you double the input light, you get double the output light. But as you go up higher and higher in illumination density, that stops happening. And the reason is you start saturating the raising of essentially exciting the fluorophore to the excited state. You have a finite excited state lifetime, and you can't get more than one photon out before that decays. And as a consequence, this then starts rolling over. And so it means if you create a sine wave that you illuminate with at high intensity, it's no longer linear, and so it'll generate harmonics. Just like if you take not just a principal wave, but you add second, third, and fourth order harmonics, you can build up higher and higher frequency information. And if we look at that, here's a dye where we crank up the intensity, and we can see not just the primary frequency, but more and more harmonics being formed as a consequence of saturating the excitation light. And each of these harmonics then gives you access to further and further and further information out about your sample. And as it turns out, there's no theoretical limit to what resolution you could actually get with a light microscope. So despite aspects of finite numerical aperture and limited wavelength, it turns out you can get access to infinite amounts of information. The problem is one practical problem of signal to noise, photo destruction of the fluorophore, those sorts of things, but not an underlying physical limitation. And so we can see that where conventional microscopy gives us access to this sort of region of Fourier space and information about the sample, linear structured illumination doubles that very nicely by creating a pattern that is the finest thing we can fit through the objective lens. By doing nonlinear structural illumination, either by the saturation method or with photo switchable dyes, we can get an arbitrary number of harmonics. It's just, again, as I said, it's a signal to noise issue and a dye issue of how far out you can go. Now, as you imagine, as you go further and further out, you're getting a smaller and smaller region of all the information because your porthole, your viewport into that area, is always the same size. It's always the same size as in the conventional microscope. But as you go further out, you need more angles to be able to fill this in and get a very nice view of what the space looks like. But with just 2 new harmonics, now recording 8 directions instead of 3 directions, you can do a very good job of filling up Fourier space to high resolution. And here's just an example with fluorescent microspheres where you can see you go from a conventional image to the linear structured illumination image to the saturated image, and you can see you get 46 nanometer, 50 nanometer resolution, and in principle this can go on and on and on. And so the problem with super resolution then becomes one of optimal dye development and better detectors, for example, and not one of fundamental physics. I want to thank the many, many people who have been involved in various aspects of this work over the years. This has been a wonderful collaboration between my lab and John Sadat's lab at UCSF. And I especially want to thank Mats Gustafsson, the late Mats Gustafsson, who is really the inventor of a number of these super resolution technologies. Thank you.